I will explain a small story about the mechanisms of apoptosis. This is a cell, mitochondria, nucleus, endoplasmic reticulum, and other ribosomes, etc. Assume it as a cell, and this is mitochondria. Mitochondria, it has double membrane structure, and cristae consists of protein cytochrome C. Its double-walled membrane has ATP. Normally, the proteins inside the mitochondria will not permit, as well as the cytochrome protein stays inside this mitochondrial space. It means the cell is alive. Whenever the protein leaks into the cytoplasm, it is the reason to initiate the mechanism of programmed cell death, apoptosis. See here, this is the outer membrane of the mitochondria. It has certain proteins like BCL2, BCLXL, MCL2, which do not allow the cytochrome C to go into the cytosol or cytoplasm. This will work until the survival signals like growth factors enters. In the case of lack of this survival signals, all these proteins will not work properly. In BH death domains, it will activate, and it activates the channels BAX and BAK. These are the channels that allow the proteins Once the cytochrome C permeable out into the cytoplasm, it will bind to the apoptosis activating factor 1, and this complex again binds to the inactivation of caspase 9. Which activates the caspase. Once this complex binds to this inactivation of caspase 9, it will get activates. This is the intrinsic one. Let's see about the extrinsic pathway. In extrinsic pathway, the cell possess death receptors like TNF receptors and FAS receptors. To bind with these receptors, there is a FAS ligand on T cell. After these two interaction, this will bind to the adapter protein like FADD. It means FAS associated death domain. This adapter protein, once activated, it binds to the inactivation of caspase 8, which is initially it is inactivated. Once it gets binding with the FADD, it will activate caspase 8 or 10. These are the intrinsic and extrinsic. Up to activation of caspase 8, it is vary in mechanisms. These are the initiation phases. In execution phase, both extrinsic and intrinsic pathways follow similar mechanism, that is execution phase. Once the activation of caspase 9 in intrinsic and activation of caspase 8 in extrinsic, they will both activate individually, not combinedly. Individually activates caspase 3. Once the activation of caspase 3 occurs, it cleaves the inhibitor of DNase, which then DNA degradation occurs, and then nuclear disintegration also occurs. From this, after nuclear disintegration, a small part of the cell, which consists of all cell organelles, is divided and it removed as the apoptotic body. It fragments as the apoptotic body, and this apoptotic body, which contains receptors that provide signals to the phagocytes. Phagocytes will come and eat this apoptotic body without leaking these intracellular contents. Because there is no leakage of these intracellular contents into extracellular space like in necrosis, there is no elicit of inflammatory actions occur here. This is the main difference between the apoptosis and necrosis. This is the whole story about the mechanism of apoptosis. Hope you will understand. Let's start with the notes on apoptosis. This is the introduction part. Like, apoptosis is a type of cell death that is induced by a tightly regulated suicide programs in which cells destined to die activate intrinsic enzymes that degrade the cell's genomic and nuclear and cytoplasmic proteins. Apoptotic cells break up into plasma membrane bound fragments like this, called apoptotic bodies, which contain portion of the cytoplasm and nucleus. So here also apoptotic body has portion of nucleus and cytoplasm. And this here in this line, plasma membrane, nothing but this remains intact. It will persist. There is no leakage of intracellular contents. Its surface membrane altered so as to produce find me and eat me signals for phagocytes. So the plasma membrane gets altered and the receptors on it send signals to the phagocyte to eat me or find me. Then phagocyte come and eat the apoptotic body. This is the part of when the apoptosis were recognized. It is in 1972 in worms. Causes of apoptosis. Two mechanisms, one's physiological and other pathological. Once you get understand on the story of the mechanism, this is all a notes on apoptosis which you can easily understood. This is a physiological situations when apoptosis occurs. The removal of supernumerary cells during development is nothing but when more cells are there, excessive cells are there when then required, then apoptosis begins to maintain the constant level of the number of the cells. Next, involution of hormone dependent tissue like endometrium removal during menstrual cycles, cell turnover in proliferating populations. It is inactivation of lymphocytes in bone marrow and thymus, like B lymphocytes, it gets removed, which is not activated, and elimination of potentially harmful self-reactive lymphocytes. It means it is to prevent immune reactions against one's own tissue, and death of host cells that have served their purpose, like neutrophils in acute inflammatory response and lymphocytes in immune response. These are removed by apoptosis. In pathologic conditions, during DNA damage, like once the DNA gets damaged, there might be DNA mutations in the cell and further modifications will occur. Before that severe thing takes place, apoptosis get activated and accumulation of misfold proteins in endoplasmic reticulum. To remove those abnormal misfolded proteins, apoptosis starts during infections. Once the cells infected with the viral infections, it is also removed by apoptosis. This is the overview of morphology. In necrosis, there is a swelling of cell, but in apoptosis, there is a cell shrinkage, which means cell size reduced, cytoplasm is dense and eosinophilic, more tightly packed of organelles. So chromatin condensation will occur, then formation of cytoplasmic pleb. In H and E stained tissues, cell appear as round or oval mass of intensively eosinophilic cytoplasm with fragments of dense nuclear chromatin. The absence of inflammatory response can also make it difficult to detect the apoptosis by light microscopy. Because there is no leakage of contents of the cell, so there is no inflammatory reactions happens here. And this is the mechanism we already discussed as like a story. Apoptosis results from the activation of enzyme called caspases. It divided into two phases, initiation phase and execution phase. There are two distinct pathways, mitochondrial pathway and death receptor pathway. This is the whole in mitochondrial pathway means it is because the mitochondrial organelle is inside the cell, it is intrinsic, and death receptors that is from outside the cell, that is extrinsic pathway. This is the overview of mitochondrial intrinsic pathway. The whole story we explained in starting, that is enough to understand this mechanism. And these are the proteins that present on the outer mitochondrial membrane. These three serves as anti-apoptotic and this pro-apoptotic, and these are another pro-apoptotics which regulates apoptosis initiators like BAD, BIM, BID, PUMA, NOXA. This is the overview of nodes. You can see this diagram. In viable cells, that means the cells which is live. Here is a survival signal like growth factors which comes and it leads to the production of anti-apoptotic proteins like BCL2. BCL2 which means it does not allow the cytochrome C proteins to leak outside the cytoplasm. So there is no leakage of cytochrome C. In apoptosis cells in which there is lack of survival signals and due to irradiation it causes DNA damage, activation of sensors like BH3 only proteins. It is the antagonisms to BCL2. Once the BH3 is activated, this is the activation, it leads to the activation of these BX, BAK channels which permits the leakage of cytochromes and other proteins. It initiates the caspase, apoptosis. You can see here both in mitochondrial intrinsic and extrinsic pathways. Once the mitochondria release the cytochrome C and here initiation of caspases. Up to initiation of caspases, these are different mechanisms of intrinsic and extrinsic pathways. It is initiation phase. In execution phase, there is same pathway. It occurs. It leads to breakdown of cytoskeleton, removal of cytoplasmic fragments of cytoplasmic pleb, apoptotic bodies, which phagocytes.